in the last video we have discussed about the central dogma and the process which was involved in the central dogma. That was transcription. Alright. What was transcription basically? Transcription was the formation of RNA by using DNA. Alright. So today we are going with the second process which is involved in the central dogma, which is translation. As you can see here the topic, translation. Now basically what will happen in translation? Alright, in translation protein will be formed by using RNA. Now as we have discussed in the previous video as well, we were having three types of RNAs. What was those three types of RNAs? Let's see here. So first RNA which we were having, that was rRNA. It was the first type which was formed by using RNA polymerase 1. Alright. So second type of RNA we were having, which was mRNA. Alright. It was formed by RNA polymerase 2. And the last type of RNA we were having, which was tRNA. Which is the transporter RNA, which was formed by using RNA polymerase 3. Alright. So as you can see here, we are having three types of RNAs. What those three types of RNAs? The first one is mRNA, not mRNA but rRNA. The second is mRNA, which is the messenger RNA. The third is tRNA, which is the transporter RNA. So rRNA was made by using polymerase, DNA polymerase, not DNA but RNA polymerase 1. Then mRNA was made by using RNA polymerase 2. Then tRNA was made by using RNA polymerase 3. Alright. As I taught you in my last video as well, that mRNA was not made like as it was made in the bacteria. Alright. Initially hRNA was made, then that hRNA was converted into this mRNA by three processes. What were those three processes basically? The first one was keeping, which was done by using methyl gynotriphosphate. Second one was tailing, which was done by adenylatic acid. Third one was on splicing, in which we cut the introns and exons were remaining, which were forming the protein. Alright. So here I am making the structure again. So let me change the color. Alright. So basically this is my mRNA. Here I am making. This is the cape. As we did this cape, with what we did this cape? We did this with methyl gynosine triphosphate. Alright. So this was done by using methyl gynosine triphosphate. Then here we did tailing. Here we did tailing. As you can see here, it was done by using adenylatic acid, this is adenylatic acid, which we use for making the tail. Alright. And this one is what? This is the mRNA. Alright. Which is carrying information about the amino acids. Alright. So this is, we have the structure of mRNA, messenger RNA. As the name suggests, it is the messenger RNA, which will give message to any other RNA for bringing the amino acids. Alright. So it is having all the information about the amino acids. Alright. it is having all the information about the amino acids. Alright, so now let's see in this picture. As you can see here, this is an mRNA, which is the messenger RNA. Alright, it is having all the information. As you can see here, these are the nucleotides. Alright, so it is having all the information about the particular amino acids. As we know that amino acids are moving freely in the cytoplasm. So these are moving freely in the cytoplasm. Alright, so what it will do, it will give message to the tRNA. Alright, so as you can see here, these are the ribosomal RNA. As you can see here, wait, let me change the color. Alright, okay, so as you can see here, ribosome, the last subunit. So these ribosomes are made of what? These ribosomes are made up of basically rRNAs, which is the ribosomal RNA. Alright, rRNA. When so many rRNAs combine together, they form the ribosome. Alright, so now ribosome does what? Like, what is the work of ribosome? Alright, so ribosome just makes the peptide bands between the amino acids. Alright, we know that. Basically, I'm making the structure here of the protein. So we know that proteins are made up of what? Proteins are made up of amino acids. Alright, so basically these are amino acids. So the bond between them is what? It is the peptide bond. Alright, peptide bond. So this peptide bond is made by the help of these ribosomes, means these rRNAs. Alright, so now let's see what's the work of tRNA, transporter RNA. So basically I'm making here the tRNA. As you can see here, tRNA is folded like this. Alright, so what this tRNA will do, tRNA will come here and it will interpret this whole code, or it will read this whole code. This will read the first 3 code first. This is the codon basically. So what it will do, transporter RNA will be coming, it will be reading this, it will be interpreting this codon, and then it will go to the cytoplasm. Then it will, what it will do, it will take an amino acid from the cytoplasm. From the cytoplasm it will be taking amino acid. Alright, so amino acids will be attached to it. Alright, now basically this is an amino acid. Now it will carry this amino acid here, and here it will start making a bone. Like, all the transporter RNAs will be carrying amino acids according to these codons. So these amino acids will be combining here by the help of peptide bond, which will be formed by the rRNA. Then this protein will be formed. So now we are just going to see with the help of diagram, which I'll be making. Wait, let me move it down. Alright, what I told you basically. Wait. As I told you, this is what? This is mRNA, which has a cape of methyl gynosine triphosphate. It has a tail, alright, and it has what? It has information about the particular amino acids. Alright, so it has information about the particular amino acids. Alright, information. Okay, so this is basically what? This is, we have mRNA, which is the messenger RNA. Now it is in the cytoplasm, because we know that transcription was happening in what? Transcription was happened in the nucleus of the cell. Alright, this is basically nucleus in which transcription was happened, means the RNA was formed here. Then it was fall down from this. So transcription was happening in the nucleus of the cell, and now translation will be happening in the cytoplasm of the cell. Alright, so this is the cytoplasm where translation will be happening. Alright, but in the case of bacteria, what was happening there? So basically transcription and translation were happening in the same component, because we know that bacteria are the prokaryotes, they are not having proper nucleus. Alright, so their transcription and translation were happening on only one component, while talking about eukaryotes, transcription is happening in nucleus of the cell, while translation is happening in the cytoplasm of the cell. Alright, now this mRNA is where? This mRNA is in the cytoplasm of the cell, because we know that now the translation will be happening in the cytoplasm of the cell. Alright, now I'm going to make rRNA, which was made by the help of RNA polymerase 1. Alright, RNA polymerase 1 made what? It made rRNA, which is the ribosomal RNA. Now I'm just going to make here the ribosomes. These are ribosomes. This is ribosome, which is the largest unit, because we know that the upper one is the largest unit and lower one is smaller unit. So here we have an other ribosome. So these are basically made up of what? These are made up of rRNA. These are made up of rRNA. Alright. So rRNAs made what? It made ribosome. And here we have mRNA. Now we have to make what? Now we have to make tRNA. Alright. I'm going to make tRNA here, which is the curved, which is folded like this. Alright. So this is basically what? This is basically tRNA, which is the transporter RNA, which is doing work of transportation. Alright. So this is transporter RNA, which means tRNA. Alright. Now it has a codon. Alright. It has a codon. Or suppose here we have the codon, which is A, wait, let me write here, U and G. Alright, AUG. Now what will be happening? tRNA will come. Wait, let me move this. tRNA will be coming and it will start interpreting this codon. Alright, it will read this codon and then again it will go to the cytoplasm. Here we know that in the cytoplasm, what are freely moving? Here, for suppose, here are the amino acids. Alright, amino acids. So here it will go to the amino acid. Now there is an interesting thing. Alright, in amino acid we have anticodon. Alright, so anticodon is where? Anticodon is on the amino acid. Like here I have one amino acid. Here its anticodon is what? For suppose here the codon is A. So A complementary is what? It's U. Alright, because we know that thymine won't be coming now. So U complementary is what? A. Uracil will be making pair with adenine. Alright. So G will be making pair with what? G will be making pair with cytosine. Alright. So C would be there. So here is what? Here is the anticodon of this codon. Basically this is the codon. Wait, let me write here so that it can be more clear. This is codon of amino acid, particular amino acid. Alright. This is the particular amino acid. Wait. This I have codon of amino acid. So this is what? This I have anticodon of amino acid. Alright. So I'm just going to write here anticodon. Alright. So this is so when tRNA will be interpreting it, it has that same amino acid codon will be 2. Alright. It will read will have the same information as in the codon, alright? So in the codon we have AUG, so here we have the AUG as well. Alright, so tRNA will go to this amino acid, it will search it out. Alright, we have so many amino acids, alright? It will be interpreting now. Now it will get an amino acid with anticodon of this, alright? UAC, which is the anticodon of AUG. Because we know that A will be making pair with uracil, adenine will be making pair with uracil, then uracil will be making pair with adenine, alright, adenine. Then last, gynosine, gyne, which will make pair with what? Cytosine, alright, cytosine. So here is the anticodon. Now what it will do, this tRNA will take this along with that, alright. So this amino acid will be attached to this now. Wait, let me make it here. So here I have amino acid, alright. Now here I have amino acid. Now it will go again to this, and here will be our amino acid then. Basically I should make this with another color so that it can be visible to you. So this I have one amino acid. So this is the codon of which amino acid? Because we know that we have 20 types of amino acids, alright. In some books there are 21 too. So we have basically so many types of amino acids, alright. So the first amino acid is always methylene, alright. So this is the codon of methylene, AUG, and anticodon we have, that is UAC, alright. So now it has an amino acid. Now here we have another codon, alright. So basically I don't remember the anticodon of another amino acid, but wait, let me do it randomly, alright. So basically I have C here, A here, then again I have U here. Now what tRNA will do again? tRNA will be interpreting it, it will read it, and it will write it, okay. Then it will go to the cytoplasm. The cytoplasm, amino acids are present. Then a particular amino acid will be having anticodon of this U. Then anticodon of U will be A. Then A, U would be there. Then again we have U. Then again A would be there. Alright, so that would be anticodon. Then tRNA will take that along with it. Amino acid will be attached to the tRNA. tRNA will bring this there. Then again we'll have this amino acid here. Alright, this amino acid we'll be having here. Alright, so now what this RNA will do? What's the work of this RNA? Now this rRNA, means ribosomal RNA, will be making peptide bond between them, because we know that between the amino acids in the protein we do have peptide bond. Alright, so this RNA will be making peptide bond between them. So basically this long chain would be formed here, amino acids, again peptide bond, again amino acid. So here basically protein will be formed, alright. So here basically what will be formed? Protein will be formed. These are amino acids. These are amino acids. So here we have the chain of amino acid which will be forming what? Protein. Alright. So we need to know the basic structure of amino acid, because as we know that protein are formed of what? They are formed of amino acids. Alright. So we need to know the structure of amino acids. So let me make it here. Wait, let me erase this so that I can make here. So in the mid we have C, carbon we have in the mid. Alright, here we have what? Carboxylic group, COOH. This is the carboxylic group, and in the center we do have C carbon. Here we have carboxylic group. Here I should write carboxylic So here we have carboxylic group. On the left hand side we do have amine group NH2. Alright. So wait, let me write here. Amine group. Alright. On the upper side we do have hydrogen, and on the downside we do have R, which is the radical. Alright. Now the question rises, why do we have different types of amino acids? Alright, as I told you, this is the basic structure of amino acid. So wait, let me tell you. So in all amino acids we do have this structure is same. Like in the mid we do have carbon, then on the left hand side, on the right hand side we do have carboxylic acid, on the left hand side we do have NH2, which is the amine group, then on the upper side we do have hydrogen. So this is seen in all amino acids. What we have the change in only in the R, only in the radical. Alright, in some cases there is methylene, in some cases there are in other amino acids. Alright, so just because of this R, just because of this radical, we do have different types of amino acids. So today we are done with the protein formation. So the formation of protein by the help of RNA is what we call the translation. Alright, so this was all about translation. In the next video we'll be going with the DNA replication. Alright, so basically this is all about